The history of human nutrition and material science has effectively been a history of extraction. For millennia, the equation has been simple and brutal. If we wanted leather, we raised a cow for years, feeding and watering it, only to slaughter it for its skin. If we needed insulin to treat diabetes, we harvested pancreases from slaughtered pigs. If we desired the binding properties of egg whites for baking, we relied on chickens. In this traditional paradigm, the organism was a black box. It was a complex, inefficient, and ethically fraught biological factory that we could breed over generations, but never truly optimize at the molecular level. We are now standing at the precipice of a fundamental inversion of this model. We are moving from an era of extraction to an era of creation. This shift is being driven by the convergence of two distinct but increasingly intertwined fields: precision fermentation and artificial intelligence. This is not merely about making fake meat or cheaper cheese. It is about the digitization of biology. It is the dawn of algorithmic fermentation, where the metabolic pathways of microscopic organisms are tuned with the same precision as a computer chip, and where functional proteins are designed not by evolution's blind trial and error, but by the predictive foresight of deep learning models. To understand the profundity of this shift, we must look beyond the buzzwords and trace the life cycle of a single protein in this new bioeconomy. Let us imagine a novel enzyme designed to break down plastic waste, or perhaps a hyperstable casein protein for a lactose-free cheese that melts perfectly. In the old world, discovering such a protein would take decades of bioprospecting, searching through rainforests or soil samples for a happy accident of nature. Today, it begins with a prompt. The journey starts in silicon. The first step is the hallucination. Using generative AI models, similar to the large language models that power chatbots, scientists can now speak the language of proteins. Proteins are essentially strings of amino acids, and their function is determined by how these strings fold into complex three-dimensional shapes. Tools like DeepMind's AlphaFold and the University of Washington's Rose TTA Fold have effectively solved the protein folding problem, allowing researchers to predict a protein's shape from its sequence. But the newest frontier is de novo design, which literally means from new. Instead of asking an AI to predict the shape of an existing protein, scientists use diffusion models, which is the same technology behind AI art generators like Midjourney, to hallucinate entirely new protein structures that have never existed in nature. For our hypothetical plastic-eating enzyme, the AI explores a vast latent space of chemical possibilities, generating millions of candidate backbone structures. It then uses inverse folding algorithms like protein MPNN to calculate the exact amino acid sequence required to create that structure. In a matter of hours, a single graphics processing unit can do what millions of years of evolution failed to accomplish. It designs a catalyst, purpose-built, for a modern problem. Once the digital sequence is finalized, it must be translated into biological reality. This is step 2, the genetic upload. This is where synthetic biology takes the baton. The AI-generated amino acid sequence is converted into a DNA sequence, optimized for a specific host organism. Typically, this is a yeast like Comagataea lefaufii, or a fungus like Trichoderma ricci. This isn't a simple copy-paste job. The AI must optimize the codons, which are the DNA words, to match the host's preferred dialect, ensuring high yields. This digital DNA code is sent to a DNA printer, synthesized chemically, and then inserted into the host microbe using CRISPR-Cas9 gene editing. The result is a cell factory. This is a yeast cell that has been hacked to prioritize the production of our novel protein above its own survival instincts. From there, the engineered microbe is moved to step 3, the digital twin. The microbe enters a bioreactor, which is a steel tank where the fermentation happens. But this is not your grandfather's brewery. Modern precision fermentation is overseen by digital twins. A digital twin is a virtual replica of the fermentation process that runs in parallel to the physical tank. Sensors inside the real reactor stream real-time data, including pH, dissolved oxygen, temperature, and metabolic waste to the digital twin. Machine learning algorithms analyze this stream, predicting how the microbes will behave hours in the future. If the AI predicts a drop in productivity due to a slight shift in acidity, it autonomously adjusts the feed rate of sugar or alkali in the physical tank before the problem actually occurs. This predictive control can reduce batch failures by up to 60% and slash energy consumption, which is a critical factor in making these products cost competitive with traditional agriculture. Finally, we reach step 4, harvest and purification. The fermentation broth, which is a soup of yeast cells, and our target protein is processed. The cells are separated, and the protein is purified. Because the target protein was designed by AI to be hyperstable or easily separable, the downstream processing costs are often lower than with natural proteins. The result is a pure, functional white powder. It could be a plastic-eating enzyme or a cheese-making casein, born of code and built by biology. The implications of this technology extend far beyond the laboratory. We are witnessing a restructuring of the global food and materials economy. This is part 2 of our analysis, the economic singularity, or the post-cow economy. The most significant barrier to the adoption of fermentation-derived proteins has always been cost. A cow is, admittedly, a very efficient bioreactor. It eats free grass and produces complex proteins. Beating that efficiency with steel tanks and electricity is difficult. However, the techno-economic landscape is shifting rapidly. Recent analysis from 2025 suggests that optimized facilities, driven by AI process control and higher yield strains, are approaching a 90% reduction in production costs compared to early 2020s pilots. We are seeing unit costs for precision fermented dairy proteins projected to hit the $8 to $13 per kilogram range. This is the parity point. This is the moment when it becomes economically irrational for a food giant not to use the fermented option. Consider the case of industrial enzymes. Companies like Anasense have reported a 10,000 times reduction in the computational cost of protein design. By bypassing expensive wet lab trial and error with zero-shot AI predictions, where the AI gets it right the first time, the R&D overhead for new biological products is collapsing. Furthermore, while the broader food tech and plant-based investment sectors cooled significantly in 2023 and 2024, precision fermentation has remained a bright spot. In the first quarter of 2025 alone, fermentation startups defied the downturn, capturing nearly half of all funding in the alternative protein sector. Investors are realizing that this is not a consumer fad like a veggie burger, it is a business-to-business infrastructure play. Companies like Liberation Labs and Synonym are building the server farms of biology. These are massive contract manufacturing facilities that will allow any startup with a DNA sequence to scale production without building their own factory. This biomanufacturing as a service model is the biological equivalent of Amazon Web Services, and it promises to unleash a Cambrian explosion of independent protein companies. Moving on to part 3, the regulatory frontier. Technology moves fast, but bureaucracy usually moves slow. Yet, in the last 18 months, the regulatory dam has begun to break, signaling a global acceptance of algorithmic fermentation. In September 2025, a landmark moment occurred when the French startup Verlet received a no-questions letter from the US FDA for its precision-fermented beta-lactoglobulin, which is a major whey protein. This effectively grants the product GRAS status, meaning generally recognized as safe, clearing the path for mass-market commercialization in the US. This was not an isolated event. The FDA has been processing a backlog of GRAS notifications, with companies like Vivici and Remilk also securing regulatory wins. The Better FDA Act 2025, a bill proposed in the US Senate, aims to make this GRAS notification mandatory rather than voluntary. While adding a compliant step, this ultimately legitimizes the industry and removes the stigma of self-regulation. Europe is also experiencing an awakening. The European Union, historically cautious and sometimes hostile toward GMOs, is showing signs of fear of missing out. Precision fermentation technically utilizes GMOs, though the final product contains no genetic material. The Netherlands, arguably the global heart of ag tech, broke rank in late 2025 to become the first EU nation to allow pre-approval tastings of fermentation-derived novel foods. This code of practice allows companies to test their AI-designed proteins with real consumers years before official EU approval. It is a tacit admission that Europe cannot afford to let the US and Singapore dominate the future of food. Meanwhile, Singapore continues to operate as the world's regulatory laboratory. The passage of the Food Safety and Security Bill in January 2025 formalized the category of defined foods, creating a streamlined, predictable pathway for high-tech proteins. By treating food security as a matter of national defense, Singapore has created a regulatory environment where AI-driven food safety assessments are welcomed rather than feared. Perhaps the most sci-fi application of algorithmic fermentation lies in its convergence with personalized medicine, which brings us to Part 4, hyper-personalization, or food as software. We are moving toward a future of hyper-personalized nutrition, where the feedback loop between our biology and our food is closed by AI. Imagine a consumer named Jane in the year 2030. She wears a biosensor that tracks her glucose spikes, cortisol levels, and specific amino acid deficiencies. She also submits a monthly microbiome sample. An AI model, trained on vast datasets like NHANES, but fine-tuned to Jane's unique physiology, detects that she is deficient in a specific branched-chain amino acid and that her gut lacks the enzymes to properly digest gluten. Instead of buying a generic multivitamin, Jane's subscription service sends a request to a local fermentation hub or perhaps a countertop appliance in her own home. There, a specific strain of yeast is induced to produce a custom protein blend rich in the missing amino acids, along with a specific enzyme to aid her digestion. The product is fermented, purified, and delivered to her door as a personalized morning shake. This is not hypothetical. Research published in 2025 regarding large language models like Mistral 7B, analyzing dietary constraints, has already demonstrated 91% accuracy in interpreting free-text dietary needs. When combined with the programmability of fermentation, food becomes software. It is versioned, updated, and patched to fix bugs in our human operating system. However, despite the optimism, the road to an algorithmic bio-future is paved with significant hurdles. This leads us to Part 5, the challenges. First, we have the black box problem. AI models like AlphaFold are often black boxes. We know that they work, but we don't always know why. When an AI designs a protein that looks nothing like anything found in nature, safety becomes a complex question. How do you prove a protein is non-allergenic if it has no biological precedent? This interpretability gap poses a challenge for regulators who are used to comparing new foods to existing ones. Second, there is the carbon cost of compute. Training the massive models required for protein design is energy-intensive. A single training run for a state-of-the-art model can emit as much carbon as 5 cars do in their lifetimes. Furthermore, while fermentation is more land-efficient than cattle farming, it is energy-hungry. Bioreactors require constant temperature control and aeration. If the electricity grid powering these cell factories is dirty, the environmental gains of animal-free protein are diminished. Finally, there is data scarcity. While we have billions of protein sequences, we have relatively little data on how those proteins actually behave in a fermenter. We lack the functional labels, meaning the data that says this sequence produced high yield at 30 degrees, but failed at 32 degrees. This data is proprietary, locked away in the servers of private biotech firms. Breaking these data silos without compromising intellectual property is the next great challenge for the industry. In conclusion, we are leaving the Neolithic era of biology, where we domesticated plants and animals, and entering the Paleotechnic era, where we domesticate the molecule itself. Algorithmic fermentation represents a fundamental decoupling of nutrition from agriculture. It promises a world where we can produce the key nutrients of milk without the cow, the whites of eggs without the chicken, and entirely new proteins that evolution never had the time to invent. It is a future where a famine in one part of the world doesn't wait for a harvest season to be solved, but can be addressed by beaming a DNA sequence to a solar-powered fermentation farm. It is a future where our food is as smart as our phones, and where the boundary between the digital code of silicon and the genetic code of carbon is finally, irrevocably, erased.